Hello, and thank you for joining us. I'm Gwen Taylor, Senior Developmental Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled The ABCs of ADC, Antibody Drug Conjugate Characterization. This webinar is being co-sponsored by Current Protocols and Waters Corporation. Waters Corporation creates business advantages for laboratory-dependent organizations by delivering practical and sustainable innovation in the areas of liquid chromatography and mass spectrometry, two vital enabling technologies for today's state-of-the-art analytical laboratories. A leader in antibody drug conjugate analysis, Waters' fit-for-purpose UPLC, QTOF-MS, and informatics software offer a solution to biopharmaceutical organizations' challenge to characterize these complex, heterogeneous structures. Current Protocols has been in continuous publication for 28 years and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 17,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your question will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days. We will send you an email with details of how to access the recorded webinar, along with a PDF of the slides and the certificate of attendance. And now it's my pleasure to introduce today's speaker. Dr. April Zhu is a senior principal scientist at Pfizer Biopharmaceutical Science with expertise in analytical development and characterization for biotherapeutics and vaccines. Following her postdoctoral work at Cornell University, Dr. Zhu joined Regeneron Pharmaceuticals in 1998, where she worked on analytical method development and protein characterization through managing a protein core facility utilizing various bioseparation techniques and mass spectrometry. She became a principal research scientist and a group leader in vaccine research analytical development at Pfizer in 2006, with a focus on method development, qualification, and characterization for early-stage vaccine projects. Since 2009, her work has focused on analytical development and bioprocess and product characterization for antibody drug conjugates. So let's go ahead and get started with a very warm welcome to you, Dr. Zhu.
Thanks, Gwen. Thanks for giving me opportunity to discuss what we have learned over the last several years on our analytical work on antibody drug conjugate characterization. This is very briefly my talk outlines, and I would like to give a little bit of background about ADCs. And I would like to spend a few minutes to talk about the challenges we encountered for ADC analysis. And then I would like to spend most of the time talk about the analytical approaches for key quality attributes determination and characterization for ADCs. And I will give specific examples on how we do analysis on cysteine chemistry ADCs, lysine chemistry ADCs, and a little bit of touch upon site-specific ADCs. And in the end, I will give a summary. And what is antibody drug conjugates? And antibody drug conjugates are typically referred as a guided missile. It takes advantages of antibody with a high potent cytotoxic drug. So advantage of this that antibody can bond with target antigen on the cancer cell surface and then takes the small toxic drug in. So once it's inside of the cell, it can cause the targeted cell death by releasing those toxins in the cell. There are a lot of advantages of using these techniques to make a drug, but in making it, there are some requirements of how you can do that. One is you need an antibody and with targeted on the abundant target expression in cells, and then the other thing is that you need a drug which is high potent, of course, and of course you have to link this drug to the antibody, and the linker actually needs to have ability. It's stable in a normal situation, but it can release the drug when it's enter into the cell surface. because of that advantages, and many companies are working on this new molecular modality. As you can see in this slide, we started with back in 2000 just to have one product from Marotac, which still product in Japan. And then in 2011, we had Citrus coming out and then Cysteine. And then right now there are over 50 candidates in the pipeline for enter into clinical trial, and 10 are in later phase 2 and 3. To understand how complicated to do the ADC analysis, we need to understand a little bit how these ADCs are made. And I'm showing here to make ADCs, you need the target antibody, which is shown in the colored picture here. Sometimes you can even have a single chain antibody. You can have a variety of the forms for antibody part to bond with antigen. Then you need to have the payload, which is shown in the bottom of the slides, which you look for classic linker payload used for ADC making. Right now, a lot of companies are developing new different linker payload with different mechanisms of actions. And to link those linker payload, you have to also have to choose different linker. So through the ways we can release it in a cell. And besides those complications, you also have to worry about where can you link the drug? How do you link the drug? Which is the chemistry is how you can put the drug on the antibody. So in this slide shown that back in probably last over two decades, most chemistries are using these two top ones are shown here. One is the lysine chemistry, one is cysteine chemistry, and the lysine chemistry is genetic and non-specific, so that you can typically have a distribution. And the classic cysteine chemistry conjugation, you're reducing the disulfide bonds between the light chain, heavy chain, and heavy chain. And then you're adding the drugs through those interchain cysteine residues. You're typically giving a profile like in the right side of the spectrum. And in recent years, because of the heterogeneity associated with conventional ADC chemistries, more effort was focused on adding the drug into specific sites, targeted sites. So advantage of this, of course, needless to say, it's less heterogeneous. And another benefit actually is more on improving the pharmaceutical properties. So because of that, most companies right now working on the last technology. But for the most of the ADCs in the clinic, I just show you that the majority of them are still made of lysine chemistry and the consistent chemistry. That's why I'm going to spend most of the time talking about the analysis of these two ADCs. And what are the analytic challenges in ADCs? I want to start off with this Venn diagram. Analytic challenges, of course, is combined from heterogeneity from that, and also having linker and the drug. And what I have is ADC is, easily to see that it's a lot more complicated than map and the linker payload. And showing on the right side is just a figure of heterogeneity in maps back in 2000, I think early days, 2005, 2004, Nature paper. You can see the map itself is already very highly heterogeneous, as most of your audience pretty much already knew that. And this is only a simplified version, and nowadays we have a lot more heterogeneity associated with the map, which needs to be characterized. And on top of that, now we have to worry about what chemistry we add on. Associated with each chemistry, they comes of certain type of heterogeneity. And in addition, one thing we need to also worry about is the drug and the linker heterogeneity. Even though most times small molecule is not that complicated as a large molecule, but a drug and the linker itself can also have its own stability issues like hydrolysis degradation, because the linker is designed to be able to release it upon the internalization in the cell. So, what are the things we have to worry about for the antibody drug conjugate? And for the map, typically, we have a certain number of quality attributes. Because this is a talk of ADC, I'm only going to focus on the quality attributes of ADCs beyond the map. But again, that for map itself, they have typical quality attributes, the size heterogeneity, charge heterogeneity. But, for ADCs, the sum of the map quality attributes are still important. And very important ones you can see that is antigen bonding, that you need a map to be able to bond with target antigen. And another type of quality attribute we sometimes need to worry about is called effective function, which map itself has the function, and we need to characterize it. And beyond the quality attributes for map, for ADCs, we typically worry about drug-to-antibody ratio. That means how many drugs are added into the antibody. And then the drug load distribution, how the drug is distributed through the antibody. And then how much free drug left out of the process, or how much drug is degraded once it's conjugated into antibody, because that's a safety concern. And the cytotoxicity is the functional assay, which means how potent is your ADC can kill the cancer cells. And then we worry about the identities of the ADCs, that's a regulatory concern. And also, when we add those drugs into the ADC, where do they add it in sites of conjugation? And there are some more of the quality attributes. Not all of them are critical, but a lot of the most critical ones are just listed here. And in the early stage, we typically set up a set of testing methods to analyze these ADCs. And showing those black ones are typically those we always test for map itself. And for the ADCs, again, we always have assays which are more targeted for ADCs. And they're showing here are highlighted in red. And for example, drug loading and drug profile typically are done by HIC, which is hydrophobic interaction chromatography, UV, imaging camera electrophoresis, or reverse phase HPLC. And the identity is typically done by peptide mapping or some other approaches. And the impurities, free drug and free antibodies, and the biologic activities, which are cytotoxicity. And for early stage, we typically comes with, different ADCs coming from different chemistry, different linker payload, different target. So there is very difficult to have one single method or even platform method for ADCs. So what we typically do is that we take the platform method for MAP. For example, for size exclusion assay, we use that for MAP. If ADC it works for ADC, we take that for ADC assay. And for those quality attributes linked to the conjugation reaction, we typically use we call the toolbox approach. For DARs, we have a set of methods available for DAR determination, regardless if they are different chemistry, different linker payload, just, one of them typically works. And for drug load profile, we typically have HIC, immediate capillary electrophoresis, mass spec, and free drugs, we can use ELISA reverse phase. Unconjugated map, we can, borrow from the drug load profile and the potency assay. And the conjugation sites, we always use in the peptide mapping by LCMS. And I just would like to give you a couple examples on this toolbox. For example, for DAR, for UV, if we want to use the UV method to determine the DAR, typically your ADC has to have a certain spectral profile. So for example, the red shown here is when you have drug load 0, which is a MAP, it's a typical protein absorbance. And for the ADCs, you typically have some drugs. In this case, you see the drug absorbs around 250-ish. So you can use the difference between MAP and ADC, calculate how many drugs on the protein. You can also done this by a reverse phase HPLC method by reducing the ADCs out. If you have an ADC reduce with certain reducing reagent like a DTT, you can have the map and forming light chains with no drug when no drug conjugate, and those with one drug conjugate, you can see the light chain with one drug. And you can have heavy chain, same thing. A lot of times the heavy chain has more drugs. You see the heavy chain with no drug, heavy chain with one drug, heavy chain with two drugs, heavy chain with three drugs. You can calculate the average drug to antibody ratio based on this profile. You can also calculate it based on the Higgs, which is hydrophobic interaction chromatography. In this case, that you have the drug load 0, drug load 2, 4, 6. They can be resolved on this method, and then you can actually do the calculation based on this chromatogram. And then if we don't have any method available, typically we use this LC-MS method running on the reduced ADCs. And in this case, I show a comparison of MAP versus ADC. In the MAP case, you just have a light chain with no drug, but in the ADC case, you have a light chain with one drug, light chain with no drug, and with heavy chains, again, the heavy chain with no drug, heavy for ADC, you have heavy chains with 0, 1, 2, 3 drugs. You can again calculate the dots on this. And to making sure that all the method actually is doing a good job, we typically do a comparison in the early stage. In the first left side of the histogram showing that the dot comparison by UV, by reverse phase HPLC, and by Higgs. You can see that dot determined by stream method are highly compatible. Again, in the right side, we put in the LCMS data in. If you comparing the dot determined by LCMS, Higgs, and the reverse phase HPLC, they're also highly compatible. In this slide, I've shown that, how we do the drug load profile determination. Typically, for the conventional cysteine chemistry, we can resolve different drug-loaded species from Higgs. In this case, again, you see that you can drug 0, 2, 4, 6, and actually last peak is 8. You can have them resolved. You can use it to look at the percentage of each drug-loaded species. For the lysine chemistry, sometimes it's a little bit difficult, and we typically using ICE method, which separates based on the charge. And you can see the different drug-loaded species most time and separate it out, and the last one is by mass spec in the bottom to separate the different drug-loaded species out. And I will spend a little bit of time in the next of my talk about details of their separation and analysis. So first, I want to go through I think probably the largest class of ADCs, which is done by conventional cysteine chemistry. Conventional cysteine chemistry ADCs are made by partially reducing the interchain disulfide bond. In this case, it's light heavy and heavy heavy. So after the reduction, the drug is added into this interchain cysteines. So if you reduce and then followed by conjugation, you can actually result into heterogeneity as shown as in the right side. For example, if drug load 2, you can have drugs added into the heavy chains. You can have drugs added into between light and heavy. And drug load 4, you can have actually 4 different isoforms. That's what we call positional isoforms. In the drug load 6, you can have potentially 3 types, and the drug load 8 is 1. So with that heterogeneity, if you run them on the Higgs, this is an example of one of the ADCs we have. You actually can resolve the drug load 0, drug load 2, and drug load 4. You actually have 2 different isoforms can be isolated in a drug load 6. So in this is assay we call a multi-attributes assay. We can use this method to determine average DAR, which is drug to antibody ratio, and we also can determine the drug load distribution and tells you what percent of drug load 4, drug load 2, and drug load 6. And we can, between 4 and drug load 4, we can tell about the isoform information, and we can determine the unconjugated map by the first drug load 0. And to start that, with a lot of time, a lot of characterization has to come with it. When you separate a Higgs without a label, you really don't know which peak is what. So we had to do some characterization before we can actually, confirm that the peak identities for us to do the calculation. So the first step typically is by collecting each peak and then to figure out what are the drug load. In this case, shown that if we collect each fractions, we run the analysis for one of the analysis, which is shown here on the right side is reverse phase for reduced map. You can see that if we take the peak fraction 2, F2, we actually can measure a 4 peaks, which is drug load 0 with for light chain, and drug load 1 with light chain, heavy chain with 0, heavy chain with 1. We can calculate based on this stoichiometry and actually obtain the drug load 2. For the same token, we can see that the drug load 4A, 4B comes out with drug load very close to 4, and the F6 comes out very close to 6. So we can this way to confirm that those peaks actually indeed is drug load 2, 4, 6. And a classic ways of calculating, also to understand the position isoforms is to collect these peaks, not only subject them into the reverse phase HPLC analysis for the reduced fractions, also run them on the CESDS. And between these two, because without the disulfide, those things, on the denature form, you're falling into light chain, heavy, and the light. I'm not going to go through details, but this is in the literature how to do this. Bottom line that you can actually understand each peak, what are the position isoforms in addition to the drug load. And nowadays, when moving on doing this much quicker, is that we can employ a two-dimensional method to do this type of analysis. And this is a collaboration work with Waters Incorporation. What we do is that we isolate each ADC. Each ADC in a heterogeneous mixture by the first dimension, which is Higgs. Then we take the fractions of Higgs, directly subject them into the second dimension. In this case, is the reverse phase HPLC method. And by doing that, we don't have to collect this peak. It saves a lot of time. Also, make the analysis a lot more efficient as we don't encounter the loss and the sample change. And by doing this, you can see that if we take a fraction, for example, taking some of the material from the one just highlighted in this rectangular, you can see that we came out with the peak 1, and the peak 1 was identified as light chain with 1 drug by mass spec directly showing in the bottom. And then peak 2 showing is a heavy chain with the 3 drugs. So you can actually easily piece back your original ADC. You know now you have a ADC with the light chain with 1 drug, heavy chain with 3 drugs, and which perform the dimer, you have the actually drug load 6 of the species. So this is just to give you a full summary of a 2D analysis. And if this is again what I mentioned is a collaboration with Waters, and it's actually done with the ADC mimics. So the Higgs profile looks slightly different from the last one I showed you, but nonetheless it's again separating different species from the Cysteine chemistry ADCs. So if you take the first A row, you see that for a dark 4 species, and when we in the first dimension Higgs, we take the peak to the second dimension, we actually can form the two different species. One is the light chain with 1 drug, one is heavy with 2 drugs with disulfide linked in the heavy chain portion. So you can see that you have the disulfide isoform, which is drug is added between the light and heavy in the chain disulfides. And for the same reason, you can do the exact same for every peak. And for drug load 6A, you can see these 2 combination of these 2 isoforms, and with drug load peak 6C, and we can see the isoform of drug load 6 and then this disulfide isoforms. And with this, we can then analysis much faster, also is much more conclusive, and we actually can do this analysis probably in 1 day rather than used to be several weeks. And using the HIC, we can actually also looking at monitor the conjugation process. In this case, we generated 3 ADCs. One is a low cysteine conjugation, one is intermediate cysteine conjugation, one is a high cysteine conjugation. You can see that in the low cysteine conjugation case, you have very low 6-8 species. In the high cysteine conjugation, you have much higher 6-8 condition. Much higher 6-8 conjugation, and you can see that the average drug load calculation from the right side of the table, average DAR is 2.8 in the low case and close to 6 in the high case. And we can also analyze this cysteine chemistry by LC-MS. I want to mention that, doing this type of analysis is really non-trivial because because they don't have the interchain disulfides to hold them. So the ADC analysis by regular LC-MS for this type of ADCs are quite difficult. So this is typically done by using a native LC-MS, where the size exclusion is HPLC is run in a native mobile phase instead of in organic and acidic phase as most LC-MS requires. And nevertheless, this can be done even though it's a little technical challenging. You can see that for this cysteine ADC chemistry, we actually see that you have map with no drug, map with 2, map with 4, map with 6. So, it matches with the HIC analysis. Again, for those ADCs, we collaborate with Waters. You can actually compare the drug load between the HIC method and between the native LC-MS analysis. You can see that the table, the DAR determination by HIC, and for 3 batches of ADCs, and then the DAR determination based on native LC-MS, they came out very good. And you can actually use either method to determine the DAR and the drug load profile for this kind of ADCs in some of the ADCs. And I just want to mention that not all the ADCs are working. You have to really testing them out. And another typical analysis we done for the ADCs, we call it is 3-part subunit. In this case, we're using an enzyme called IDS. We cut below the hinge region of the map. What it does that when you cut it, it becomes the 2 parts. But if you reduce the disulfide bond, it becomes the 3 parts. So it results into a single chain FC light chain with no drug and the FD prime region. In this case, that we can actually current mass spectrometer, we be able to match each domain with the isotope resolution. And for the ADCs, in addition to the single chain FC, light chain, the FD with no drug, because the drug added, some of them added the light chain 1 drug, and some of them added the FD domain with 1 drug, 2 drug, and 3 drug. You actually can resolve all them out. And actually with 2 drugs, you can resolve the positional isoforms as well. And the last analysis for cysteine chemistry is typically do is peptide mapping, and that's done for different purposes. A lot of time was just the identity, as I mentioned, and also we typically also use this method to confirm the size of conjugation. Because the linker payload is hydrophobic, they typically show up in the last portion of the HPLC chromogram as I circled here. If you're comparing to the map with the ADC, you can see that, the peak I put a arrow there, that the map is a lot taller because that is specific size is conjugated. That way, you're in the ADC case, the non-conjugated peptides much smaller. Same is true with the other peak here. And those peaks were converted with drug-loaded peptides, which helps you to figure out where it adds and how much it adds. And now I'm going to switch to talk about some analysis done for the lysine ADCs. And the lysine ADC is actually just by non-specifically adding the linker payload into lysine residuals of the map. In any typical map, there are over 80 lysine residuals in the map. And because of that, so it's a lot of difficult to resolve those heterogeneities out in lysine ADCs. So for the star determination typically we done by UV for the linker payload has a specific absorbance profile. I'm showing a case of where one of the ADC we have is that you can see the map is a typical, in the middle is a typical UV profile here. And for the linker payload, it absorbs, looks pretty similar, but it does absorb beyond the map, which I show in the red vertical line. So when you form an ADC, it absorbs in both ways. We can actually calculate the linker payload portion based on the extinction coefficient of the linker payload and subtract map at 280, the contribution. We can actually calculate the linker payload concentration, protein concentration, then calculate the DAR, which is the drug to antibody ratio. And a lot of ADCs can be done that way. I can say that also for cysteine chemistry, sometimes this can be also a much useful method and then faster or robust method than a HIT. And just I mentioned that heterogeneity is in the lysine ADCs. And this is, if we separate the ADC by the Higgs hydrophobic interaction chromatography, as you see in the traditional classic cysteine chemistry case, you can resolve the different drug-loaded species 0 to 0, 2, 4, 6, 8. But here it's really difficult to resolve them. In addition, because it's random adding the lysines, you can have drug load from 2, 3, 4, 5. 6, 7. Out of each 4, 5, you can have the isoforms as well. And nonetheless, we can sometimes still use Higgs to help to understand, to help with process development. And showing on the right side, just showing that if you really collect these Higgs fractions, you can see that they indeed correspond into different drug-loaded species, as the case in the drug. First, peak 1 has no drug, and the peak 8 actually. Peak 7 contains most of drug 8 and the drug 7 species. And for drug load profile for the lysine, as I just briefly mentioned earlier, sometimes we utilize the charge separation. In this case, showing that if you have a map which show in the middle, there's a typical charge heterogeneity of a map. You have some very small basic species and a lot of acidic species showing in the lower PI region. When you're adding drug in which showing in the ADC, you see that you don't have any of the map anymore, and most peaks convert into species eluding migrating in the lower PI region, and that's because now you're adding a drug. You actually can sort of estimate how many drugs there are based on the species shown on the top, starting from 0, 1, and 2. You can see its first peak may start from the drug load 3 and beyond. And another method is we're using mass spec, and this you just resolve intact mass. I must say that using mass spec typically is a little bit more challenging than other techniques. Typically, unless we have to, we don't use this. Although nowadays mass spec has become more powerful and more user friendly, a lot of people can now run them routinely. For this ones, we actually, have to have some challenges because in this particular ADC, there is a hydrogen bond, which is acid labile. And again, we have to use the non-TFA, which is, has to be, pH has to be higher to run this acid, more close to relative condition to get this distribution. In a summary of the drug load profile by the lysine chemistry, you can see that we can have 3 methods, but each method has its own caveat. And for the Higgs method, you can clearly see that not well resolved. And for ICE, I want to just make a note here. They are indeed complicated with the charge profile of map itself. As you just recall, just to speak for map itself, it always comes with a small percentage of acidic species and basic species that can be co-eluding with each drug-loaded species. So this can be only used for monitor the product, but you cannot say any species differently 100% drug-loaded species. For the LCMS method, even though this is no doubt, it tells you the integrity of your ADCs, what species you have, but the caveat is that it does suffer the electron spray efficiency heterogeneity, because sometimes high drug-loaded species doesn't have the same efficiency as the low drug-loaded species. Nevertheless, we always use one of the methods to use for drug load profile. And for lysine chemistry, this becomes more important to determine the size of conjugation. And again, using the same peptide matching method, we can achieve this goal. But for lysine chemistry, it's a lot more complicated. We use the lysine and digest, and lysine cuts after the C-terminus of lysine. But if the lysine is conjugated, they cannot be cleaved by this enzyme. So what typically, we can do is look for the new peaks. Sometimes they co-eluding, so you really have to do a mass spec analysis. Thing is, we typically look is what are the peptides that affected, which is, again, by arrows showing here, for example, you see this peptide is in ADC is dropping the intensity. And by doing this type of analysis, we can actually also estimate what percentage of that particular residue is conjugated. Because of this, all these caveats, with the work we have done with the Waters Corporation, and we have worked on a new ways of, mapping those lysine sites. In this one, we not only using the trypsin using lysine digest, we sometimes come with trypsin digest to make into a much smaller fragments. And for the aspartame digest, which we comes with is a non-lysine digest, the trypsin or the cuts of lysine. With aspartame, it's cutting, before the aspartate. And if we do that, and the advantage is that we actually generate just one single peaks, and you can, out of the comparing to the lysine digest. And following for mass spec analysis, we really can, identify the sites and can also calculate abundance. And showing in this slide is for the 3 ADCs we collaborated with Waters. And you can see that for that 3 particular ADCs, we can identify all the sites which are conjugated. In addition to that, we can calculate the site occupancy, what percent of sites was occupied. And you can see that, in the green is the ADCs which was more conjugate, has higher level of conjugation, and red was intermediate, and blue is the lowest one. You can see that one of the sites here actually is highly conjugated, but they corresponding to the level of conjugation we mentioned earlier. And using this approach, we believe you can actually get better information than just with lysine alone. In the last few minutes, I want to talk about analysis for the site-specific conjugation. And the site-specific conjugation, the field right now is really expanding a lot, and there are multiple conjugation chemistries used, and approaches used are not only, right now, early days was engineering a cysteine into the map, so let's make that available to conjugate the linker payload. And now they can putting a non-natural amino acid into it, and you can engineering other hydro tags into it. And another classic of ways is using enzymatic mediated approach using just transglutamase and adding the linker payload to the glutamine residues by engineering, again, some tags into the map. So now I want to list that with this, as I discussed, the ADCs does have less heterogeneity. The final ADCs has less heterogeneity, and they are easy to analyze. And most methods I just talk about, if you look at it, actually can be simpler to adopt into analysis of this. However, I want to mention that, there are significant amount of analytical work may be needed for the map portion, because this map now no longer traditional platform map. When you add in a cysteine, engineering a cysteine, engineering a tagging, and engineering, do a non-natural amino acid, and there are a lot of more analytical work needed to making sure your map is made properly. And for the ADCs, a lot of times with, for example, cysteine mutant using cysteine mutation approach, that those cysteines a lot of times directly out of the cells cannot be directly conjugated. So there are a lot of more additional process work needed, and therefore a lot of analytical work needs to go with it to analyze those cysteine mutation ADCs. And there are more things, you know, can discuss. This can be easily a 30-40 minutes talk, but I think, you know, we will wait until we have in the future anytime. But you are welcome to ask any questions. It's just to show you that how simple it can get for the, if you do the peptide mapping on the site-specific ADCs, and now you have just one peak out of the conjugated peptide, and now on the map used to have a peptide, now is no longer there in the ADCs. In the last point I want to touch upon, there are two additional key analysis needs to be done for ADCs. One is a free drug analysis. And free drug, again, is, you know, it can form from two ways. One is that just a residue-free drug which isn't cleared from process. The other can come from the product. Once you purified it, and your product can start degrading if you're not installed them properly, you also have to measure them. And clearly there are important safety concerns because the free drug is typically highly potent. You want to make sure you are keep them in a well safety, below safety concerns. And nowadays, a lot of industry are done by reverse phase HPLC, showing on the left side just the two ADCs. If you look at the reaction mixture, you actually can see that you have a lot of free drug and related species. When we purify it, we can purify them really low, down to almost typically well below LOQ of the master. And again, in the bottom part, again, in this case, you can have everything purified. And another analysis we typically done is the functional assay, biologic assay, which is the ADC cytotoxicity, and just to making sure that this assay is to ensure that your map can bond with the cell and then can release the linker payload and then kill the cell. So this is typically done by using a cell lines expressing the antigens of interest. And in this assay, showing that you have to show that if we at a high concentration of ADCs, you can inhibit the cell growth, which is induced sometimes a lot of it is done by through the cell killing, induce cytotoxicity of the cells, and then when you dilute on the ADC, they maintain actually no inhibition in the cell. So that's by that, I want to actually conclude my talk, and then just hopefully I've shown you a little bit of flavor that heterogeneity in ADCs does present unique analytic challenges, and I also think that's a good opportunity for analytic chemists. And then I hope I give you a little bit of tools for ADC characterization. We discussed our general strategies and also discussed how to do analysis based on different conjugation platforms, like conventional Cysteine and Lysine chemistry. And right now at Pfizer, we believe we build up extensive analytic capabilities, and we can actually support the process and product characterization for all the ADC development. With that, I want to acknowledge most people working on it, and it takes a lot of people to generate all the assets. And here in the pharma science, Pfizer, R&D, this is near and not close to a complete list. I just listed a few key players, and also the process group, which is our bioprocess group and the conjugation group, and our management and the ARD. And with that, I thank you for your attention.
All right, thank you so much, Dr. Zhu. So let's go ahead to the question and answer segment of the event. So if you haven't yet submitted a question, now is the time to do so by using the Ask a Question box at the bottom of your screen. So let's go ahead and see what questions have come in so far. All right, the first says, normally the DAR analysis of ADC from low-dose PK samples is more challenging than the DAR analysis of drug substance due to the much lower level of ADC in the PK samples. Would you please comment on which analytical method you normally use for DAR analysis for in vivo PK samples, particularly for the later time points, such as after 2 weeks?
That's a very good question. And I'm not a good person to answer that question because we don't do the in vivo. But based on my interactions with our colleagues in a different department, and we typically actually done them by LCMS.
Okay. All right, our next question says, Dear April, thank you so much for your great presentation. Could you please elaborate more on TIC analysis of the cysteine conjugates, such as which columns are appropriate for reliable TIC of ADCs?
For the Higgs separation, the column we use actually is a butyl chemistry. Higgs is non-porous from, I believe it's from Tulsa. Is a chromatography company which makes a lot of columns for size exclusion and columns, and also the hydrophobic interaction columns. Okay.
Next question asks, is there a good agreement between the drug load calculation and the biological response? Is there an analysis method that has better agreement with the biological responses?
I think that question I'm not totally understanding. Can you just rephrase it a little?
Sure. I guess the first part of the question maybe has the major part of the question. It says, is there good agreement between the drug load calculation and the biological response?
Yeah, for the first question, I would say yes. And there are literatures have done, by other companies, which is that shown that when you have lower drug, you have low efficacy. But at a high drug, even though sometimes you have the more efficacy, but sometimes we do see the safety issues. So that's why for this ADC field, the old dogma is that people are targeting at drug load of 4. That's what I believe the current 2 products in the market are targeted at that DAR number. And the recent development also in this field have shown that actually there are a lot of site dependence. And some of if you in a specific site, you actually can add more DARs, add more drugs into one specific site to achieve the better efficacy with the actual low toxicity.
Okay, and the second part of that question was saying, is there an analysis method such as HIC, ICE, or LCMS that has the best agreement with the biological responses?
Out of the 3 methods, typically, I think HIC was, if I remember, in the field, a lot of people actually looking at the retention time of the HIC of specific ATC and the correlation with actually the PK study. And I think nowadays in the field, I think some of them are in the publications, you do see good correlations between HIC and the real pharmaceutical properties.
Mm-hmm. All right, next question. How do you identify those ICE peaks of the ADC?
That's a very good question. It is a little challenging because the typical ICE method doesn't allow you to isolate the peaks out and for the characterization. For the work we've done here, you show in my presentation, we actually took the peaks from HIC. Because if you can see that HIC had given some resolutions, we took the HIC fractions out of the HIC separation, then we subject both of them to mass spec and ICE, then to get the correlation of the peak. I didn't show every slide just for the sake of time. We do have done that by separating them out of the HIC. And I think in literature, people also using the preparative scale of the capillary electrophoresis method to isolate fractions out.
Okay, on to the next question. It says there are so many cysteine isoforms of ADC. Do you know if any of them are more potent or more stable than others? That's
a very good question. With isoforms, typically, we don't have the ability right now to separate them in the prep scale, but the answer is that we don't know the answer. But in reality, when we look at the heterogeneity, the heterogeneity of the whole mixture, we actually believe that isoforms probably does not differ that much if they have the same drug load.
Okay, on to the next question. So how easy is it to link HIC separation with LCMS analysis of the DAR species? Presumably some desalting is necessary prior to LCMS. Yes,
so that's what I discussed here. Characterization of Higgs fractions, it can be a tedious. It can take tedious amount of work, and what we typically do in the back though is collect the fraction, then desalting, concentrating, then subject them into the further mass spec. Showing in my 2D setup the work we done with water the corporation, we actually do them online. So right now you take the Higgs fraction and directly subject to reverse phase, which is desalting them online right away, and then there's analyzing the reverse phase by the mass spec. So that's a more preferred way if you have a system set up, or you can purchase a system. Right now, both Waters and other vendors also making a dedicated 2D system for this kind of purpose.
Okay, there's actually a second part to that question that I didn't read previously, but it is it also feasible to link HIC with intact ADC LCMS analysis?
That's a very good question, and that's something we always trying to do. But right now, I don't think technology is there. The potential is there, but technology is not there yet, because with if it's a traditional if it's with a traditional lysine chemistry with a stable linker, if you have the sensitivity, it should be easily done. But with the conventional cysteine chemistry, that's even harder, because the things has to be done in a native conformation. And right now, I don't think the mass spec for intact analysis sensitivity in the native condition is there yet.
Okay, on to the next question. Asks, what type of HPLC methods, which includes mobile phases and columns, would you recommend for antibody separation?
What kind of columns, right? And for us, typically because monoclonal antibodies are quite a large size and hydrophobic, and when we use the reverse phase analysis, and for reverse phase, we typically choose at least the C4 column, or we sometimes choose the CN column, CN chemistry, cyanochemistry. And for the mass spec analysis nowadays, most people are actually using the size exclusion used just for desalting before the mass spec analysis. If you don't want to resolve a lot of species out of the map, just have mass spec to do the characterization to measure the species out.
Okay, on to the next question. It's actually a two-part, so I'll just give you the first one first. It says, what have you found to be the biggest challenge with this method for cysteine-linked ADCs?
I think the biggest challenge with conventional cysteine ADCs actually is analyzing those positional isoforms. And with others, should be actually things are easier because they can be separated by Higgs, but positional isoforms are typical ones that cannot be measured by conventional methods. So, but I think at least recognizing in the field, and that's why I don't think if you really do drug development, right now they're not requiring you to provide those information yet. But it is something for you to understand, for you to particularly control your process.
Okay, the second part of that question was what amount of ADC is needed to get a good signal for the SEC LCMS?
Well, a second LCMS right now, a good amount is, I think if you have a 10 microgram injector with current mass spectrometer, any current state of the art, the top Q-top, it should be good signal. And sometimes you can get away with even lower amount. Of course, if you have more material than 10 microgram, it should be even better.
Okay, on to the next question. It says, can you elaborate on how to quantify residual drug in an ADC?
Yeah, and for the residual drug in ADC, it actually can be quite challenging. The reason is that we are typically looking for very low amount of the drug in the presence of large amount of proteins. So typically, if you just do the direct column separation, it's a little bit difficult. So typically, we have to either by precipitating protein out, then measuring a small drug, or by using a certain columns which actually does not bond the protein but bond with small drug, and then analyze them, quantify them by typically by something similar to a reverse phase HPLC.
Okay, on to the next question. It says, for conventional cysteine and lysine chemistry ADCs, each ADC has heterogeneity with a distribution of different DAR species. So have you looked at how each DAR species affects the efficacy and the safety of the ADCs?
I think in the field, people have done those kind of work. There are some literatures that are shown that, similar to a question we asked earlier, that shown that the drug loaded DAR species, if it's lower, typically it's less efficacy, but it's safer. But when the drug load is higher, it's more efficacious, but it can have higher toxicity. So it's definitely a balance, but it's not true for every ADCs. It basically, if you're working on certain ADCs, you probably still have to do this type of study to understand the effect of DAR on the safety and the efficacy.
Okay, we have time for a couple more questions here. The next one is, do you see mass spec being employed for QC of ADCs?
I think actually that's a very good question. I think the field can be, that's the future direction has been moving on to use a mass spec to do the QC release. And I think for ADCs, since if we have a free drug assay by LCMS, that can see potentially it can be used for in a QC setting, and some of the DAR measurement, certainly for if reverse phase, for reduced map, and followed by mass spec, either a direct mass spec, those all potential be able to be released by QC.
Okay, on to the next question. It says, if multiple methods exist for DAR and drug distribution determination, how do you choose one for analysis? Which one is the best?
And typically what we do is that to see which is the most simple one. For example, if we have the DAR determination, if there is a UV method available, and we'll typically go with UV. That's because it's the most reliable, also the simplest method and most robust method. If there is no, those methods available, we look into which one is simpler and which one is easier to be, especially in the pharmaceutical industry, will be more, be able to release by QC in the downstream.
Okay, I think we have time for maybe two more. This next question says, how important is it to know the drug to antibody ratio?
That's I would say is the most important quality attributes you have to know for any ADCs. And the reason that is both the efficacy concern and the safety concern. And DAR typically is something telling you both by both things, we also, how good process you are, how many drugs you added on. That depends on, you wouldn't anticipate how much drugs actually will be dosed into patient, and that's certainly one of the first thing you want to measure when you make any of the ADC.
Okay, on to probably the last question here. It says, how important are the data analysis software tools for ADC characterization, and what's available and what might be missing?
For ADC, there are some tools available for helping you to calculate the R. For example, if you have good chromatography method in some other software, in like Waters, they have a software to help you input, identify the peaks with the DAR numbers, and then directly, give you the output with the DARs with the drug load profile. And for I see that for the software concern, and a lot of times in the industry, they derive some certain information, but not directly measuring them. Sometimes for unconjugated map, they derive the data based on the mass spec, and those part in the software does need to. There are some works needed for software development because it's not obvious there. Especially sometimes how do you correlate drug load profile with the process parameters, and those are the fields may need some work.
All right, so we are now out of time, and so we're going to conclude the question and answer session. I'd like to let you know that today's webinar has been recorded and will be available for on-demand viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with a PDF of the slides and instructions on how to personalize and print a certificate of attendance. So on behalf of today's speaker, Dr. April Zhu, and our sponsor, Waters Corporation, we sincerely appreciate your attending today's webinar, and we look forward to your attendance at future events from Current Protocol.